Morning talks going. Excited to have, starting with Archishman Raju. He's here at NCBS and he's going to talk about some very exciting stuff on Waddington landscape, which I've particularly enjoyed a lot for many years.
Okay, thank you, Sid. And yeah, so I'll be, talking about some of the work we've been doing for a while on Waddington landscapes. And I guess I just put these two pictures just to show you a few other illustrations of the landscape, as I'm sure many of you have seen the classic picture in the Waddington book, Strategy of the Genes. But to start off with, well, I should also say thank you to the organizers for the, I should have said it first, I'm sorry. But, I think this reminded me, I do think that this is a topic, this question of, high dimensional spaces is a topic that doesn't just require new methods and new techniques to understand data, but I really think requires new theoretical effort to really try and make sense of data that we now all agree is available and is very, very high dimensional. So to start off with, I just wanted to, I'm going to talk primarily about developmental systems. And mostly I'm going to talk about cell fate, how different cells in your body take different fates. Here is just a UMAP image. These kinds of images we've seen a lot of now. This is the developing C. elegans embryo. And the only reason to put this image is to say that even though we are dealing with high dimensional data, I think almost everybody agrees that there is some low dimensional representation of that data. And the very reason that we look at these kinds of images and draw them in low dimensions, right, has an implicit assumption that there must be a low dimensional representation of the data. Otherwise, these images would make no sense. But of course, a lot of the way that this kind of compression is done is sometimes heuristic. And I think it's a theoretical challenge to come up with ways to really dimensionally reduce the data in ways that are sort of theoretically justified. Okay, so with that introduction, I guess thanks to Marianne, who sort of introduced some of the beginning concepts that I want to mention, which is that if you actually look at trying to model cell fate decisions, then you immediately encounter these very, very high dimensional models, because you have these, for example, signal transduction networks, which you could have some ligand binding to a receptor, but then downstream of that is some, in this case, MAP kinase cascade, which has many, many components involved. And the more we seem to study these things, the more every single signaling, like the Wnt signaling pathway, has hundreds of components. So many, many, many components seem to be involved in even basic cell fate decisions. In this case, the cell just has to decide whether it's going to proliferate or differentiate, but that involves really many, many components that are part of that decision. And if you actually try and write down a model for this, so you try and take these components, you write differential equations for how they're going to evolve in time, and you come up with some right-hand side, then, of course, first of all, I should mention that we have no real fundamental way to write down these right-hand sides. I mean, I guess if you wanted, you could really try and model the chemistry, but most of the time what people do is they use phenomenological equations, Hill's functions, and so on, which are not really things that we have tested, but are sort of our guesses for how these components evolve in time. And then further on, if you take these kinds of models and then you look at the sensitivity of the models to the parameters, which is what Marianne mentioned in the morning, then right now there's been a long series of papers which show you that this sensitivity has this sloppy spectrum, that there are only some directions in parameter space that matter and other directions that don't matter. I should just say that it's not obvious what those directions are, which is why this problem is difficult, because the bare parameters may all seem like they matter, and it's not obvious how to resolve what those directions are, and in some sense that is an outstanding challenge. Okay, so cell-fate decisions from the point of view of a single cell seem to be very complex. But on the other hand, you have this observation that they're both robust, and we have a few hundred types of cells in our body. So there's some discrete types of cells which long work in developmental biology has classified. And they're pretty robust. If you make perturbations, you look at mutations over expression experiments, you don't start getting new cell types. Typically, what you start getting is just permutations of known cell types. So you may convert one type of cell to another type of cell with a perturbation, biological perturbation, but you typically will not start creating half a skin cell and half a neuron or anything like that. So they're quite hierarchically organized, and they're quite robust. The types of cells are quite robust to these perturbations. Yeah. Yeah. Right. Yeah. So I'm going to mention a system with a hybrid cell also in a bit. So that's true. It's not. So this is why not always true, and you can in cases get these kinds of hybrid phenotypes. Yeah, but I'm just making the sort of broader statement here that this is the typical cases that you just permute between known cell types, not necessarily the. Yeah. 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 Okay, well, that's a good question, and I guess it really goes to the definition of a cell type, right? And how do you, I mean, there are various ways you could identify a cell type, right? So do you want to look at transcriptomic data? Do you want to look at it physiologically? And I think that's sort of a longer discussion, but I would say that it is surprisingly rare, or to put it another way, like if you look at this, right? If you look at this single cell data for these developing organisms, it's often correspond, maybe that's because people are trying to make it correspond to known cell types. And so, right, and we don't have a good way of clustering, so often people cluster it in a way that you get known cell types. So maybe that's a bias of the way we're doing it. But I think, yeah, I mean, so that's a discussion, but I would say that's more the exception rather than the rule. That I would say that all this long work that has been in developmental biology, that's the main, this main classification into cell types has more or less held. Of course, there's variation, around you can't really define exactly what a cell type is. But anyway, that's a longer discussion. Maybe we need to think about them in a different way. I'm not opposed to that. Yeah.
Hear me? Okay.
Yeah.
So I perhaps don't have as much background in things like this. So could you quickly just summarize what the network is trying to do, or the pathway is trying to do, and sort of what are the inputs?
So this is the only network I'm going to show you. So we're going to forget networks after this. So
okay.
Let me skip that. Yeah. Yeah, that's an interesting question. I think so. And obviously it's been shown to be able to influence, you can very strongly influence fates of cells. Yeah, and I think that's actually an understudied question, the extracellular. Yeah. Okay, so, the reason I titled this talk, Waddingtonian perspective, is first to say that this observation that you have these discrete cell types is something that Waddington was trying to show in this landscape, that the you can think about the cell instead of thinking it from the network perspective, which I'm not going to talk about so much. You can instead think about a landscape in which the cell, the ball rolls down, and these minima correspond to different types of differentiated cell types. And I want to mention, this is a quote from his Strategy of the Genes, where he says it is important to realize that the comparatively simple orderliness of the epigenetic landscape is a property of a higher order dependent on an underlying network of interactions which is vastly more complicated. And I think this is significant because it's clear that what he is thinking about is what we would today call, right, an emergent description. I think what he was thinking about the landscape very clearly, I think in many other places, was that the landscape is some sort of emergent description of how these cells are taking their fate. And even though there may be a vastly complicated underlying network, that network first of all, there may be many networks which may give you similar landscapes. And second, that it was possible to study these landscapes even if you don't have knowledge about the network itself. And I'm just going to take a minute. I'm emboldened by Kabir to talk a little bit about the history and philosophy of this, which is that, right, if you read a biology textbook, then you'll see, right, that there's this old debate between mechanism and vitalism in biology. This is like in textbooks, and they say the mechanists won, the vitalists were wrong, and it's right, everybody's happy. But the Waddington was not just an individual, he was part of a group of people who call themselves the organicists. And they call themselves the organicists, and they felt that they had a way to think about development, because if you look back, the vitalists were pretty much all developmental biologists. And they were saying, well, how can you get this phenomena from a mechanistic viewpoint? And Waddington was part of this organicists who thought of themselves as a third way. We are neither mechanists nor are we vitalists. We have a third way. And interestingly, I think the organicists actually went on, I think there's a clear connection, there's evidence to say that they went on to influence the way that a lot of condensed matter physicists would start thinking about how to model materials and the language of emergence and so on. Actually, I think you can trace back to biology more than you can trace back to condensed matter physics. But of course, we know that, right, mechanism wins in biology, and particularly with the advent of molecular biology, this idea, this way of thinking becomes sort of it's not pursued. But what I'm going to try and argue in this talk is that the landscape particularly is not just a metaphor, it's not just it's something that we can mathematically formalize and usefully, and it is useful to understand certain types of biological data. Okay, so the framework of how I'm going to think about cell fate is, again, I think Marianne mentioned in the morning. I'll just briefly say, right, again, so in principle, you have some set of equations, right? Let's say of gene expression with time. I'm not going to bother about what those equations are. They come from some network, they come from some interactions, who knows? But in principle, you have some differential equations which tell you how gene expression changes as a function of time. Now, there's a nice analogy between concepts from dynamical systems theory, which is sort of the right mathematical framework to think about this problem, and ideas from developmental biology. So, right, the differentiated cell type is a fixed point, is a stable fixed point. The saddle points are the interesting quantities, because the saddle points are precisely the points where the cell has to decide whether it's going to go to one fixed point or the other. The saddle points are really what are going to be most interesting. Another remark to be made is that, of course, right, I think also, as was mentioned in the morning, when we say fixed points and stable fixed points, I think you always have to remember that there are some time scales involved. And so often you will assume something is a stable fixed point for the relevant time scale. And then eventually it may become unstable and may start becoming, right, it may not, there may be a bifurcation. Who knows what will happen? But often you're modeling a certain decision in developmental biology, and for that time scale, it is fine to assume that something is a stable fixed point, even if it may not remain so in later times. Okay, so there may be some dynamic gene expression so that's very true. And then you assume that what really specifies the cell, right, or what you're interested in, the quantities you're interested in studying are the ones that reach a fixed point. But of course, there may be oscillating genes and all sorts of other genes which are not maybe relevant for this decision about whether the cell becomes one or the other kind of cell. Okay, so the way the way we've been thinking about this is that so it turns out that, and this is early work that goes back to René Thom, Stephen Smale, and so on. But it turns out that a large class of flows that we think are biologically relevant are gradient flows, which means that, right, you can really think of the landscape as something that's sort of mathematically defined, right? So, gradient flow means that it's a gradient of a potential, with the caveat that you need a Riemannian metric. And so it turns out that you can take a large class of flows. I'm not going to tell you the precise details, I can discuss offline, but just to tell you a few of the assumptions that go into this, right? You have to assume structural stability, which means that you assume these flows are qualitatively similar if you make small perturbations to the flow, and you have to assume boundedness. In that case, it turns out that you can write there's a theorem that tells you that you can write these flows as just gradient flows with the caveat that there's a metric. I'm not going to deal with periodic orbits. I mean, periodic orbits are interesting, but that's not something I'm going to deal with in this talk. So we're dealing with flows that don't have periodic orbits. Structural stability means there's no chaos, for example. And so you're dealing with a limited class of flows, but still a very, very broad class of flows that we think are biologically interesting.
Clarify, is the statement that the gradient-like flows are structurally stable, or is it that structurally stable flows are gradient-like?
Right. So structurally, stable flows plus a few assumptions are these Morse-Mail systems. These Morse-Mail systems are gradient-like. Yeah. Okay. Okay, but so this is true, and here I'm just showing you actually that here this is just a potential, and you get you have just a simple bistable potential. Here I'm just showing you what a metric can do, right? So metric here, all I've changed is I've taken a potential and I've added a Riemannian metric to the flow, and if you do that, you can get terms that look like curls, but they're no longer but this is all in the metric. So it's still the steady states, the fixed points, all of the fixed points are in the potential, but the dynamics between the fixed points is something that the metric can change. So that's from the theorem. I'm not going to talk about the metric so much. It is an interesting quantity. I'm happy to discuss about it later. But mostly, I'm just going to assume that we can take these potentials, and really, if we have a self-weight decision, we can write down a potential to describe its flow. Yeah. No. Because it's positive, so it's you can show that the metric doesn't change the yeah. If you just that's right. If you're just interested in steady state, you can ignore the metric. It's the dynamics between the steady states that the metric can change. Okay, now what we're interested in is if you make biological perturbations, if you have a mutant, if you overexpress a signal, how does my landscape change? So what we found is that you could have some underlying equation, but really it turns out that you can write it as a potential. The second thing is that, well, really you need to be worried about the cell types that you have in the developmental decision that you're concerned with, and you should model them as steady states. And then the final question is, well, but if I make biological perturbations, I can change those steady states, so how do I model that? Of course, what perturbation you make, mathematics is sort of agnostic to. It could be some concentration of a signaling molecule. If you could change decay rates, rate coefficients, whatever in principle you can change. All of that is just some parameter which is changing the structure of this landscape. So now we have a parameterized landscape with some fixed points, and which can be changed by changing parameters that you may have access to biologically. Okay, so there are two kinds of bifurcations, it turns out, that are generic, and which are the saddle node and what's called the heteroclone. Yeah.
Question. This statement that you have essentially gradient flows, is it actually a statement about those functions Fi that you had in an earlier transparency, dGi by dt was Fi of the G's? So is it a statement about the nature of those Fi's?
It's not. So it is really just a statement about the structure of the flow. It's not a statement about what those Fi's are. So a way to think about it is, if you think about near the fixed point, near the fixed point, you could linearize whatever the Fi's are, and you'll get some general matrix. What this theorem is telling you is that if you're a gradient flow, that matrix would be symmetric. And so because you have a metric, you actually have two matrices. So you can take two symmetric matrices and you can multiply them to get whatever matrix you want, which also is something you can show. So it doesn't matter what the original Fi was. You can always do this given the other assumptions: structural stability, bounded flows, and so on.
And this is not a local statement near fixed points.
It's not. It's a global statement, yeah. And the metric is crucial to show it. You can obviously in general you don't get gradient. May has to be positive, definite. I mean, whether they're biological constraints, I don't know. That's a good question. Okay, so it turns out that as you change the landscape, there are two bifurcations that are generic. One is the saddle node bifurcation, which means you have a saddle point here and a stable fixed point, and they, for example, annihilate each other. The other is what's called the heteroclinic flip, which may be a little less familiar. But the basic idea is if I have three types of cells, so I have these three fixed points, and I imagine escaping from this attractor, then the question is, I have a heteroclinic orbit here. Question is, do I go to this, to the left or to the right? And basically, if I move the positions of these saddles relative to each other, that can change whether my outset from this saddle goes to this fixed point or it goes to this fixed point. And making that change is called a heteroclinic flip. It just means that you have a heteroclinic orbit and you can flip whether things go to the left or the right by making a very small change. Okay, now in a two-parameter space, you can imagine there's a curve of bifurcation. Oh yeah, go ahead.
At the steady state, or in the saddle in that diagram, which way it goes to, right?
I'm talking, yeah, I mean, I will talk about multiple cells later on, but here you can think of this as a landscape for a single cell.
For a single cell.
Yeah, for a single cell.
And
yes.
then for the that heteroclinic flip.
Yes.
How is it different?
So you see this trajectory. You see the outset of the saddle goes directly to the saddle. That means if you had no noise, you would really go from this saddle to, you perturb exactly in this direction, you would end up here. In this trajectory, I've just changed it slightly, and now the outset goes to this fixed point. And the reason we are interested in this is that biologically, of course, you're interested if I escape a certain kind of cell type, if I'm going through my process of differentiation, will I end up on the left or the right? These will be different kinds of cells. So that's why this is biologically interesting. I want to know when I escape my attractor through whatever mechanism, I want to know whether I'll go here or I'll go to the other fixed point. Okay, now if you imagine you have two parameters, these two parameters then give you they'll give you a sort of a curve of bifurcations. So in this case, I've drawn a line of saddle node bifurcations. So again, these are I'm keeping it a little abstract here because it is sort of abstract that you could take your equations, you change two parameters in your equations where you have these, let's say, three stable fixed points, right? As you change those parameters, whatever your favorite network equations are, you're going to get these curve of saddle node bifurcations. And in this case, this means that you go from a place where you just have two stable fixed points to three stable fixed points. So that's the saddle node. And then you can also, of course, get these dashed lines, which are these heteroclinic flips, which are telling you, again, if there's a heteroclinic orbit, whether you go to the left or the right. Right. So what we've shown so far is we can model things with a potential, assuming these mathematical assumptions are biologically relevant. If you change signals, there are two bifurcations that are important, the saddle node and the heteroclinic flip. And as you make these biological perturbations, you're interested in what is the bifurcation diagram going to look like. So again, I'm making no reference to the precise equations that are governing the system. Okay, and then it turns out, and I'm not going to go into details, but you can make actually not very difficult arguments to show that there are only certain kinds of ways in which these bifurcation lines could meet each other. So as you have these two parameters that you could change, and let's say you have a system where there are three stable fixed points, basically these lines of bifurcations can either meet perpendicularly or they can meet or they can become parallel. If they become parallel, you get a cusp. If they meet perpendicularly, then they just meet perpendicularly. And then you can basically write down some classes of bifurcation diagrams that you can get, which if you had three stable fixed points, really tells you, really tells you what kinds of bifurcations you could get if you had any developmental system which has three stable fixed points. So again, this is a very powerful statement. It's saying that you take your favorite dynamical system, which has three stable fixed points, you vary any two parameters in that dynamical system, you can write down the classes of bifurcations in that you will get as you vary those two parameters. And then the interesting question becomes, which class does your dynamical system fall in? And how could you possibly distinguish between being in one class or the other class? Okay, so I'm going to give you an example now to show you sort of how this works out. Yeah. So just a minute. So I'm trying to figure out if you're saying how would you figure out what the class is, or are you saying what is the, maybe you can read, yeah. Yeah, like what? Right. 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 Okay, yeah, so that's a good. So for example, actually, if you just vary different parameters, you could take the same system and you could get a different diagram. That's in principle possible. There are some cases in which you really are. So for example, this and this really are sort of topologically different. And so in that case, you need to do a little bit more to go from one class to the other class. Yeah. Yeah, so if you have 3, it becomes harder. Yeah. So in 2, you can really say all that would happen. If you have 3, it's just you get higher co-dimension bifurcations, which are harder to, I mean, you can't sort of mathematically classify. I mean, you can in principle, but it's just harder to mathematically classify all of it. So again, the advantage is, the disadvantage of this is, of course, that you're dealing with a few stable fixed points and 2 parameters. But I will say that, right, biologically, 2 parameters is not 2. This means that, for example, if you have 2 signaling pathways that control your decision and you can vary them, that's a pretty, you know, that's a typical thing that you would do. The advantage of this is it's totally general. You're not relying on the form of the equations. You're not relying on the network. You're really saying these are the, you take whatever network you want and you're saying these are the possibilities. No, this is a mathematical statement. And in principle, actually, you could get slightly more complex diagrams, but these we think are the minimal classes. So this is a mathematical statement. There's no biology yet here. That's also a mathematical statement. That you can go from any Morse-Mail system to any other Morse-Mail system using these 2 bifurcations. Yeah. The only plausible connection to biology is, first of all, that this is going to be interesting for biological systems. And the second is that the assumption of structural stability is something that you could plausibly imagine in developmental systems as applicable, that you don't imagine that your developmental systems are chaotic. They don't go off into some crazy trajectory, but are actually, and boundedness is also something that presumably is something, right? Your biological molecules are bounded, yeah. Yes, of the original system. Yes, that's correct. Yeah. So you have a ball which you put around your fixed points, and then you can make these statements. It doesn't depend on the dimension of the original system. Yeah. Yes. If you have 4, you have other diagrams. Yeah. Yeah. That is still fine. In 4, you can still do it. It's a longer list. Yeah. Okay, so to show you how this works in an example and actually see how to tie this to a biological system, I'm just going to start off with an example from early mouse development. So early mouse development, you have, initially you have a zygote, there's cleavage, you get multiple cells. And then you have 2 binary decisions. The first one is from blastomers to what's called the trophectoderm and the inner cell mass. And the second decision is the inner cell mass going to the primitive endoderm and the epiblast. And the epiblast is actually what will end up becoming the embryo proper. The second of these decisions is what I'm going to be interested in. It turns out to be controlled by the FGF signaling pathway, which means if I give a high dose of FGF, then you get all primitive endoderm. If I treat my embryo with an inhibitor, then I get all epiblast. Okay, so this is sort of in the class of systems that I've described to you abstractly, where you have 3 cell types, at least again at these time scales. All of these are going to differentiate further. Particularly the epiblast is going to differentiate further. But at these time scales, you have 3 cell types: the ICM, the primitive endoderm, and the epiblast. You have a signal, you have a parameter that you can vary, and that, as I said, perturbs the number of cells which go into this versus the other fixed point. Okay, so before I actually give the landscape description of this, one of the first things we had to deal with is the question of, maybe it was raised earlier, the fact that you have multiple cells. And you have this FGF signal that's controlling this decision, but that means that the cell will secrete an FGF ligand outside. That ligand will go into the receptor of another cell, right? And then that cell will decide, based on the FGF expression that it sees, what fate it's going to take. So you have multiple cells in this, at this time in the embryo, you have between 32 to 100 cells. So the first thing we had to do was, well, we have this landscape at the single cell level. How do we extend it to this case where there are multiple cells? And what we did was we looked we looked at the spatial correlations in the activity of this molecule ERK, which is downstream of the FGF. So presumably its ERK expression is a proxy for FGF levels. It's something that you can get live imaging data for. And it turns out that actually there's not that much spatial correlation. So the ERK of a cell and the ERK of a neighbor, I mean, there's some mild anti-correlation, but basically there's not that much spatial correlation in the ERK expression of neighboring cells. Which tells you that basically the FGF is somehow getting homogenized, right? And that can happen through two ways. Of course, the FGF can diffuse, but the second thing is that these cells actually are moving around quite a lot. And so, these decisions are happening over time scales of a day, but these cells are changing their neighbors over, every 20 minutes, every half an hour. So these cells are moving around, the FGF is diffusing, and so you really don't have this idea of that, one cell will give, secrete a ligand which its neighbor will see and then adopt a different fate. Instead, you have this sort of highly dynamic situation where things are diffusing, cells are moving. But actually that, of course, from a physics perspective makes things simpler, because what that means is that you can do effectively a kind of mean field description of the system. It means that you can sort of average over the noise, and you can imagine that every cell really sees one homogeneous concentration of the ligand, and that homogeneous concentration is a function of the state of all of the cells, right? So you're not going to be now concerned with the FGF ligand that every cell is secreting and every cell is seeing. Instead, you're going to assume every cell is seeing one concentration of FGF, but what that concentration is can be dependent on the state of the cell. So just to give you a simple picture, if you imagine that you just have a bistable well and FGF is like a field that tilts you, and you have, let's say, 10 cells that start off and you want a 5:5 ratio, if 5 of the cells by chance fall into one of the wells, then the FGF field will tilt the other way so that the other 5 will fall, in principle can fall to the other way. So that you can get some ratio that you want. The FGF level is a function of the state of the cells, but all the cells are seeing just one field. Okay, now the question to ask, to figure out what class this decision falls in is really what is the structure of the landscape. And here in this case in particular, basically we found two structures that you have to decide between. One is this thing, which we call the dual cusp, and the other is, again, we just call it the heteroclinic flip. But the important point is that these are distinct ways in which you could take this decision to go from the inner cell mass to the primitive endoderm or epiblast. In the first case, you have two decisions. One, you either go to the left or you go to the right. In the second case, you also have two decisions, so you can look at the positions of the saddles here. But the first decision is really just to exit the inner cell mass attractor, and the second decision is choosing between these two states. So these are distinct ways to decide this, yeah. So you mean how do we actually, So what you do is, once you figure out the topology of the landscape, you basically write the simplest polynomial which is consistent with the topology. Yeah. Third direction. Yeah, sure. Yeah, I mean, yeah, the gradient, I mean, so these are potentials, so the gradient is telling you your dynamics, yes. Yeah. So we don't put a metric here. I'll show you the data that we are trying to deal with. The metric doesn't seem to be relevant. If we had dynamical data, then the metric would become relevant. That's not the data that we have right now. Yeah. Oh, the axes here. Yeah, so the axes are abstract. In this, I mean, in principle, they are some complex combination of the gene expression. But we don't, again, I'm actually going to just be modeling the cell fates and not the genes themselves. And so you never actually have to think about what the axes are because you're only dealing with cell fates. So you just have to record, do I go left or do I go right? and not exactly which genes have. If you did have genes, then for example, if you had two markers that were right, then in principle, what this is telling you is that you could try and fit the dynamics of those two markers. And so then the axis would get biological meaning. And then you would also need the metric and so on to really get that dynamics correctly. But in this, yeah. That's one way to think about the decision. But here, the axis could be some combination of 1,000 genes. Yeah. Yeah. Uh-huh. Yeah, I'll show you that. Yeah, that's a good question. Okay, so that's a, yeah, okay. Yeah, I mean, so you need, in this case, so actually, this is actually really one-dimensional. There is really one curve that takes you from here to here. So even though I've drawn two axes, this is really a one-dimensional system. This really, you need just two axes to be able to put these three fixed points. I mean, you could have it in 3 dimensions, but effectively, you would have a 2-dimensional surface on which you could fit these things. So you could have a 1-dimensional surface in which you could actually, so this is effectively 1-dimensional, this is basically effectively 2-dimensional. You unhappy? Okay. Okay, we could discuss later maybe. Yeah. I know you said right. I'm sorry. Yeah. Yes. 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 So that's the thing, you're not reducing. So in this, so far, what I mean, this is a good question. How do you go from actual gene expression to these kinds of landscapes? That's, I think, an active area of research. But here, what I'm doing is not trying to go from the genes. All I'm saying is you have your system, you have these 3 cell fates. You're going to record, as I make a perturbation, how many of one type do I get, how many of the other type I get. Mathematically, I only need 2 dimensions to put these attractors in this way. So 2-dimension is my minimal model for y for a system of this form. Actually, here I just need 1 dimension in principle to be able to capture this topology. Yeah. So I'm not, so the dimension here comes from the topology of these fixed points and not from the data, because the data I'm dealing with is not gene expression data. The data, as I'll show you, I'll be dealing with is cell fate data. Okay, so this question of, right, what is the biological difference between these two kinds of topologies? So the one way to think about it is really to imagine that somehow I bias my system so that it only becomes one. For example, I have a mutant which only becomes one type of. I mean, maybe not mutant because I want to do this biasing dynamically. So I put in, let's say, a lot of FGF, which takes you towards one of these fixed points. And then I add a lot of the inhibitors so that I have to go to the left, right? So I put in a signal that takes you towards the right. And then dynamically, I perturb it again, and I put a signal that takes you towards the left. Now, in this kind of topology, basically the only route for it is to go back and then come to this, fixed point. There is no direct, I mean, that's the sort of gradient route for it to come back. It has to go back to the original fixed point and then come here. Whereas in this kind of topology, right, if I bias things to go to the right, I have a direct route between these two fixed points so that it can come to the left. So again, you can sort of see in the potential, right, there's basically one route. I mean, all of these things are much, much higher potential. So to come back, you really have to go back to where you started from and then fall here. Whereas here you have a direct connection between, there's a saddle directly between these two fixed points. Here the saddles are between the center and the right and the center and the left. Yeah. Yeah. That's right. That's right, yeah. I didn't show them. I didn't show the bifurcations, but yeah, if you bias, you will get the bifurcations so that you go towards, so I'm here just showing you the unbiased flow diagram. And then when you do bias them, you can get these bifurcations. But still, the structure of the bifurcations is that you still have to follow the same route back. I mean, unless you do something very drastic. Yeah. Oh, I'll come to that, yeah. Yeah. Yeah, yeah. No, so that's a good question. But I would like to say, right, this is during development. Reprogramming is a slightly more complex affair because, right, you have already differentiated things that, and it's actually very, very hard to reprogram things. So it took us so long to. Here, it's quite simple. Here, right, you just have to change the signal. Things are biased. These cells are basically they're still potent. You can still just put in a signal and right, quote unquote, reprogram them. They've not yet done all the downstream changes, which actually will make it much harder to go from this fixed point to this fixed point. So reprogramming, I think, is a slightly different and more complex problem. Whereas here, during development, you actually have access to all these cell types, and you can perturb things quite simply to take you from one to the other. Yeah. Okay. So that's right. So in principle, this is something you can test. You could put in a signal, you could put in the inhibitor, and you can have your two markers which tell you whether I'm in this state, or this state, and I can just check whether my markers have to go back to the original state, or is there a direct route between the two cell types. Okay, and then this question of, right, why we prefer the flip is that now there's been a lot of work to show. Oh, I'm late. Okay, there's been a lot of work to show that it's not only that you have this robustness at the level of the cell types, in that you're not creating new cell types. You also have robustness at the population level, right? So if you take 60-40 as the wild type ratio, even if you try to mess up that ratio, you, for example, scramble. You take these embryos, you bias half of them to go to the right, and then you mix it with a wild type embryo. Then the combination will actually still show you roughly close to the wild type numbers, which means that there is some mechanism, of course, to be able to get ratios of cell types right at the population level, and so there's some robustness also at the population level. And we can't show the data is not yet good enough to show it certainly, but we do think that this kind of a topology is obviously easier to make population level decisions here than in this kind of topology. It's easier to get sort of robustness at the population level here, we think, than in this. So we took this topology and basically we tried to fit the available experimental data on this system. And I should say that despite all of these models, there's surprisingly little work to quantitatively fit cell fate data to models. And part of the reason for that is, I guess, what we were talking about, sloppy models, that if you do a quantitative fit, you're going to get something that is over-parameterized. If I take those set of differential equations that I had earlier, and I try and quantitatively fit to cell fate data, right, when I perturb a signal, what is my cell fates, I'm just going to get an over-parameterized fit. So there's, in many cases, we've now seen, even though there are these models, the models show the right fixed points, and then qualitatively things look fine, but then they're not actually quantitatively fit to the kind of data that exists. So that's something that we tried to do, and maybe in the interest of time, I won't sort of go through all of this, but just to show you the kind of data that we are dealing with, right? You can have this is the relevant time scale. It's about 2 days. You can have an inhibitor that you put in. You can have FGF4 overexpression experiments. You put in more of the protein. You can put in some of the protein, then you can put in the inhibitor, and so on. You can put in the inhibitor, then put in a control media. So these are the kinds of experiments that you could do. And what you keep track of are the fraction of cells that take one fate, or the fraction of cells that take the other fate. And then the question is, can a simple landscape model like this, where I write down the simplest potential that's consistent with this, capture sort of this kind of cell fate data across experiments? And we found that we were able to do a good job with basically, right, you have here you have sort of, I forget the exact number, but right, you have to include time scales and so on, but basically it's around 3 or 4 parameters which can capture sort of a bunch of data across different experiments. Okay, so this is to show that this abstract framework can be useful for fitting this kind of experimental data, where you have cell fate data that's biologically perturbed. And of course, it gives you ideas for the kinds of experiments that you could do to tell you more about what these landscapes are, what these systems are actually like, and what are the most interesting kind of experiments are dynamical ones, so the kind of thing that I said here, right, where you do dynamical perturbations rather than putting in, right, just overexpressing the protein and letting it be. You actually give a dynamical signal. Maybe you put in protein, you put in the inhibitor, and then this model would make a prediction for what you would observe for that dynamical signal. Yeah.
So here, when you're kind of putting in your perturbations into this framework,
Yeah.
Are they all like different kinds of pills? Like, how do you kind of parameterize a particular perturbation?
So in this case, really all of these are FGF. So there's just one signaling pathway. If there's another signaling pathway that would have been relevant here, then yeah, they're essentially just tilts the different vector directions in the landscape. Right. Which is an interesting question, right? And that's something I can discuss with you later. That actually these, in principle, have some nonlinear effect on the landscape, and that nonlinearity, I think, can have various interesting consequences, which I won't discuss here.
And so there's no possibility of creating a new fixed point given the signal, right? The fixed points already exist and building.
Which is the observation. You don't, when you put in these signals, you don't get new cell types.
On that, so I guess the cell types, as you said, like you already had the question of how do we actually define them, right?
Yeah,
So if you imagine that you had taken some 10 marker genes, right?
Yeah.
And that collapsed. There were 4, let's say, and 2 of them kind of overlapped. And you only were working with 3.
Yeah.
Would it kind of break in some way what you would interpret? Or it's kind of robust to that kind of biological error, which seems quite easy to make?
So maybe I didn't understand.
So let's say there were 10 marker genes you used, right?
Yeah.
And you actually had 4 cell types.
Okay.
And you ended up using 3 marker genes.
Okay.
Which kind of, collapsed 2 of the cell types to
into one. I see. I see.
example.
Yeah. In the, No, so this would not be robust to that. Yeah.
It
so
would
this
not
is,
be. Okay.
Yeah. So I mean, what this is doing is really relying on available experimental data on cell types and on signaling pathways. A lot of hard work that's been done in developmental biology. And saying first that you can quantitatively fit the available data, and second that you can make predictions for more interesting perturbations that you could do to the system. Yeah, it's not making predictions for new cell, because it's not a network, it's not, right? So it's not making predictions for new cell types or anything like that. Yeah. Yeah, no, that's a good question. It takes me to my next part. Well, first of all, I'm not sure. I don't think so, because it seems to me that just from the apart from the topology, you have the actual equations, and it's not at all clear to me that the actual equations you cannot manipulate in a way that takes you from one topology to another topology. But it does seem, for example, so something we've been doing with Mohit at ISC, who has these kinds of network models. And typically in the network models, you often focus on motifs, is trying to see, well, do you often get, for a bunch of range of parameters, do you often seem to have some sort of correspondence between these network models and these landscapes? But it's not clear to me there's any absolute statement to be made there, but there may be some observation that some kinds of network models tend to give certain types of landscapes versus other. And so that is something that we're exploring. But maybe I will skip this in the interest of time, because I want to go to the other part in what 10 minutes that I have. Yeah, but here again, just to say, right, so this was actually with the, this was EMT in the context of cancer, epithelial to mesenchymal transition in the context of cancer. Here there is this hybrid cell type that was discovered. But again, so what we do in this case, again, so we're not making predictions for new cell types, but you again have these 3 types of cells. And again, in this case, you have TGF beta signaling. You can have different concentrations of TGF beta over time that gives, you have prediction for what the fates are. Fates as classified by, again, 2 markers. And you can make you can sort of, again, take a simple potential, and you can fit you can fit this kind of data, is what we showed. And again, though network models were known, right, there were no quantitative fits for this kind of data. Yeah. the concentration of the signaling molecule, for example. Yeah, exactly. So here what's changing is precisely the concentration. There's external TGF-beta that's being put in at different concentrations, because of which you're getting different cell fates at different times. And that's exactly what you're trying to. Now, frequency, I mean, I don't know. So like ERK, I mean, if you have a transcription factor and that has certain frequency, I think that can have interesting effects. That's also something we're studying, but not something I'll talk about. Yeah, I can talk to you later. Oh, this is the data and the model is in solid lines. Sorry, I should have put that somewhere. 3 fixed points, epithelial, mesenchymal, and hybrid type. Yeah, so I just want to do this quickly so I could move on to the next part. Okay, so I'm just going to quickly, I don't have a lot of time, but I will just quickly say how we've been thinking about this now for spatial patterns. And in fact, you know, Waddington himself, if you see, was looking not just at single cells, but he was imagining these landscapes for multiple cells and entire phenotypes, really. And of course, it becomes, especially if you have a growth process and so on, then it becomes, it's a good question, how do you even interpret it? But if you have a certain given number of cells and you have a patterning process on them, then there is still a way in which you can start thinking of these landscapes for spatial patterning systems. In particular, we start looking at Turing patterns. I'll again go a little bit quickly, but right, Turing patterns, as many of you, maybe most of you know, is just reaction diffusion equations. The classic model is the Gieder-Meinhardt model, local activation, long-range inhibition. And what you get is instability, a diffusive instability, which produces a pattern. In this case, a bump, right? So you have a bump in the activator, bump in the inhibitor. And essentially, there have been 2 issues, I would say, with Turing patterns. One is, again, that the reaction terms are phenomenological. You have no way to write down the reaction terms in some fundamental way. The second is that typically, as we saw, right, just to repeat myself, gene regulatory and signaling networks are typically more complex. So it's been hard, though there are some cases, to figure out which molecules are going to give you this kind of activator-inhibitor structure that will produce a pattern state. Okay, so again, we wanted to go away from this and see, can we think about Turing patterns now using these landscapes? And the idea is that we have, right, again, in Princeton, we have some gene regulatory network. Who knows, it's complex. There are some diffusing elements. There are some non-diffusive elements. And it leads to some expression, right? If I measure one of the genes, they have some patterned expression in space. Can we start thinking about those patterns now, the dynamics of those patterns as motion down a landscape? And basically, in the case of Turing, really what you have to do is just go away from thinking about the single cells to thinking about, right, some basis functions, and start thinking about the dynamics of the coefficients of those basis functions. And obviously, the obvious basis function in this case is just to take your Fourier modes. So if you now just look at the dynamics of Fourier modes, those dynamics you can think of as motion down a landscape. Going to the left, maybe you have a pattern with a single bump. Going to the right, you have where you've made some perturbation. Let's say you have a mutant where this node no longer exists, and then you get 2 bumps in the expression of this particular gene that you're actually measuring in space. Okay, and the way we decided to think about this, how to model these patterns, is again using dynamical systems theory, and we used normal form theory. And the idea behind normal form theory is that, again, now I have some motion in these various different Fourier modes, the coefficients of these Fourier modes. I have some actual flow, right? So these are, let's say, the coefficients, the radial part of the coefficients. The actual flow starts off at the homogeneous state, where I have 0 coefficients for all the modes. Then it goes towards some fixed point where I have some pattern, which is a combination of, say, several different Fourier modes. But I can make an analytic coordinate transformation that brings me from this system to this system, which is the normal form, where again, I just go in the straight line from a saddle point to a stable fixed point. Now these saddle points and stable fixed points are not cell types, they're entire patterns on space, right? So this could be, for example, a bump. This would be a homogeneous state in space. And essentially our idea was that you can capture the dynamics of the pattern using the normal form, and you can capture the actual expression by using coordinate transformations. So the normal form tells you if I make all possible analytic coordinate transformations, what is the simplest form in which I can reduce my dynamics. And then my actual dynamics is, of course, going to have the normal form flow plus the corrections that I get from coordinate transformations. And what we did was to develop basically a kind of systematic perturbation theory to include those corrections from coordinate transformations. So the procedure is that I may have some gene expression I measure at different times. I sort of project that essentially onto Fourier modes to get on the unstable Fourier mode to get some trajectory, which there's a universal form for. And then I include the corrections, the predicted corrections. So we have sort of a hierarchy of which kinds of corrections you should include to reconstruct using a time-independent map. So from this normal form space, which is low dimensional, you reconstruct, right, the whole spatial pattern using these kinds of corrections. So again, I don't have the equations here and I don't have time, but I can show you how we do it mathematically. But just to show you an example where this is relevant, right, is of course there's this paper now from 10 years ago where you have this 3-component network which produces a Turing pattern. And since then, there's been a lot of effort invested in trying to think about higher order component networks which produce Turing patterns. And in this case, right, this is the limb bud, and you're looking at digit formation, so fingers in your thumb in the mouse. And you can imagine why this is an example, why people would think there's an example of a Turing pattern. It seems like basically one mode which is growing. But if you look at the actual, and so that's the sort of model that was made, you have some mode that grows and you get, right, this is a one-dimensional slice of the gene expression pattern. But if you look at the actual data, right, you actually see that you have, if you just concentrate on the SOX9 expression, which is in red here, you sort of get 1 peak, then you get 2 peaks, and then you get 3 peaks. So that's something that we had to deal with. But to begin with, we just took this Turing network and said, well, can we sort of understand its dynamics using our normal form theory? And so this actually worked very well. So we took this network, we instead of actually putting these they had these cubic terms, we put in Hill's functions, right? It produces a Turing pattern. And we're actually, you can see the shape of these patterns is sort of complex. It's not just a single mode, but we're able to capture it very well with basically our normal form model, which just requires 2 + 2 parameters. So again, this is reduction in parameter space done in a sort of very systematic way using normal form theory and an extra perturbation theory. And the idea is, again, that we are not going to use things about the original equations, right? This is just a numerical example that we did to try out our theory. But the idea is you will just have some gene expression data. And how do I write down the simplest theory which would be consistent with that gene expression spatial patterning data? And of course, the interesting thing is for large regulatory networks, right? So you can take some very large regulatory, not very large regulatory network. You can put in some diffusers. You can put in activation inhibition. You can look at how you can, give it a Turing instability, see how it develops, and you are able to capture that dynamics very well with the very low dimensional model using this kind of normal form theory. Okay, but just to conclude in my last minute, I will say that, right, we are still bothered about this problem that you actually don't, Turing predicts that you get things just rising, one mode rising, but you actually get these things coming one by one. Essentially what we realized is that if you have, right, and so something also discussed in other ways in that same paper, but if you have an external morphogen which is interacting with your Turing system, then you can get dynamics like this, things which go one by one. I'm going to skip this slide. But show you that, right, if I have a sort of wave front and I have a Turing pattern, then I have a Turing patterning system network, then you actually get things, you can get things which actually come up one by one rather than all coming up at the same time. And the reason is simple, right? The wavefront basically divides a patterning region from a non-patterning region, and as it grows, you basically start getting your patterns one by one as it moves in space. We did a little bit of work. We couldn't sort of analytically figure out how to solve this problem. Essentially, basically now what you've done is you've made your coefficients in your reaction diffusion model a function of time and space. But we were able to come up with a good approximation for what kinds of eigenfunctions, no longer Fourier modes, but different kinds of eigenfunctions that work well to capture this kind. So now I'm just showing you the SOX9 data at 3 different time points. And I'm showing you a fit using our eigenfunctions at a particular, at one of those time points. And you can basically do a good job of, it's not a lot of data at present, but you can do a good job of capturing what exists with these kinds of low dimensional models. And of course, the kind of thing this will be useful for is if I have, and in this case, it's known that the sonic hedgehog, right? If I have some external morphogen which can perturb this pattern, so SHH inhibition, for example, leads to loss of digits, and so it would be useful to try and parameterize SHH inhibition, and then seeing, right, if we can get if you had dynamics of say SOX9 expression in the presence of SHH inhibition, that our model would predict what it would look like. Okay, so sorry for going fast, and I guess I'll just leave my conclusion slide, and maybe in the interest of time, not talk about it. And thank you very much for your attention.
We can take one question, and Marianne, you can.
Last thing you said, you have two parameters for fit. Does that include the coordinate transformation that you have to do?
So that's the +2. Yeah.
I guess let's thank Archishman, and I think there'll be more time to ask questions, so please do later. Yeah.